We're live.
Well, good evening, everyone. Welcome to this latest in our series of webinars. And we host today's event under the banner of the Centre for Intelligent Design in the UK. And we're really pleased that you've joined us for this session. Our objective over these last several months has been to present a sequence of topics which provide an understanding of aspects of design in nature. And we've been particularly keen to share evidence that calls the kind of default understanding about the origin and complexity of living systems, and indeed their many characteristics, into question. And asking questions about that, considering the evidence that we've been looking at, is not only the healthy pursuit of truth here, but it's the way that the scientific understanding has developed and grown over the last several hundred years. So we started off by focusing on design and virology, and you'll find a series of 4 recordings on our YouTube channel under that heading. Then we considered the current status of origin of life science, and we've looked at various forms of bias within the scientific thinking. And most recently, we dived into the fossil record with Günter Bechley last month. And we've really benefited from the input of world-class colleagues, people like Mike Behe, Steve Meyer, Jim Tour, Doug Axe, and so on. And today, I'm really delighted to introduce our guest speaker, Dr. Paul Nelson. Paul comes from the Chicago area and is a philosopher of biology. He's been engaged in exploring the design hypothesis for 3 decades or maybe more. He graduated initially from the University of Pittsburgh with a degree in philosophy, did his doctoral work on philosophy and evolutionary biology at the University of Chicago. And he's been associated with the Discovery Institute in Seattle, where he's a senior fellow, and is in international demand, and rightly so, as a lecturer and broadcaster. He has an extensive published output, numerous articles, book chapters. Indeed, he's one of the co-authors of the textbook Explore Evolution, and is an active member of several professional societies related to his academic background. And as you're about to discover, he's a gifted and engaging communicator. I've been very aware of Paul's work for years, and I'm confident that you'll be impressed by what you hear today as he speaks to us about orphan genes and why they matter. Now, after Paul gives his presentation, I'm going to ask my colleague David Williams, who's also part of the board of the Centre for Intelligent Design in Britain, to moderate the question session. So please make use of the chat facility on YouTube to post your questions and comments as we go along. Paul, it's a real delight to welcome you to the UK, albeit virtually. Can I invite you now to go ahead and tell us about these orphan genes and why they matter? Over to you.
Okay, can you see my title slide?
Yes, we can.
Awesome. Well, it's a delight to be with you. We expect a foot of snow here in Chicago tonight. So after I'm done with you, I'll be going out to shovel. But grateful for the snow and looking forward to spring. And looking forward to communicating with all of you about this topic, which to my mind is the most interesting aspect of biology right now because it was so unexpected. So I want to start with a thought experiment. Using the notion of a remarkable library with millions of unusual books. The parallels between natural language and genetics are quite striking, and we'll touch on those throughout the talk. But this library is remarkable because every book in the library has a distinctive feature. So imagine that we're visiting this library, and this actually is a library at Johns Hopkins University in Baltimore in real life. And we go to an alcove there, and I ask you to pick a book at random from the shelf. So you do, and I say, what do you observe? So you open the book and you notice that every page has words you've never seen before. So you collect them, let's say about a dozen or so, and you say, no worries, I will take these to the OED, which is the best dictionary currently in English, because the OED, which is updated annually, has all the words. So you trot off to the OED or you call it up online. And I made up these words, by the way, here on the left. Although this one, Mechy, is actually a slang term in my family that's a combination of messy and heck. So when something is mechy, it's really bad. In any case, the rest of them are made up just to illustrate the point. And you trot off to the OED, and the OED says, sorry, stumped, no matches. Now, it turns out, as you go through the library sampling randomly, you find the same pattern of unique and previously unknown words in every book you examine. That's why the library is so remarkable. And you plot a curve. It's a rarefaction curve. And on the vertical axis here, we have R, which is the number of unique English words discovered. And you point out to me that as you plot the curve, it is still increasing with each new book you examine. Now, ordinarily, when one writes a dictionary, like the OED, the sampling curve will initially rise steeply as you are building your sample of what's out there in sequence space, but eventually the curve will flatten. And it will stay largely flat through time. There'll be little bumps as new words come in, but most novel coinages in English are actually constructed from previously existing words. So hip hop, for instance, that word describing a kind of music is a coinage of two previously existing English words. And I'm old enough to remember when the word ripoff entered the English language in the late 1960s. But in effect, the sampling curve will remain flat through time after you saturate the space. But in this remarkable library, it's not converging, it's not flattening. And you say to me, you know, Paul, I think we don't really understand the English language very well, because if you count them, there seem to be many more previously unknown words than those we have seen already. All right, with that thought experiment in mind, let's look at biology today. So the scientific universe in which we find ourselves, really from the mid-19th century until today, is dominated by Darwin. In particular, the Darwinian theory, which was, I would say, well, it's not arguably, I would say it is novel with Darwin in 1859, namely the common descent of all life on Earth, was expressed by Darwin in The Origin, first edition, in the great summing up chapter, number 14, where he says all living things have much in common, and they can be represented by a great tree that is rooted in some one primordial form. Now we can draw a direct lineage from Darwin's conception to biology in 2021. So affinities we understand mainly now through molecular data, DNA, RNA, proteins, their sequences. A great tree is the theory of universal common descent. And some one primordial form we understand to be the last universal common ancestor. So over this period, the orthodox or textbook geometry for the history of life has been a single great tree rooted in the last universal common ancestor. And I would say that that geometry was Darwin's most significant scientific achievement. Far more important, actually, than the process or mechanism of natural selection. As a student, I learned that many evolutionary biologists, such as Stephen Gould, who I got to know as a graduate student, was skeptical of the efficacy of natural selection for lots of episodes in the history of life, but he had no doubt whatsoever about the theory of universal common descent. Now, this certainty has tended to erode over the past decade and a half in the light of genomic data, and we'll discuss that in some detail later in the talk. But by and large, if you choose an evolutionary biologist, just pick one off a university faculty out of the telephone directory, he or she will accept this picture. Now, as I said, the picture is, or the geometry of universal common descent is under pressure. And it's under pressure from a technology that really was not practical until the mid-1990s, namely automated DNA sequencing. So I have, as a student of biology, I lived through this. I was a graduate student at Chicago from 1985 to 1997, with 3 years in Boston, while my wife was a medical fellow at Harvard, and 2 daughters were born during that period. So there was a lot going on in my life, but I was paying attention to the development of this technology, and it really has revolutionized biology. In particular, we now have data that were really unanticipated in their details throughout most of the 20th century. It wasn't until the 20th century was nearly over, in fact, that GenBank, which is in the United States the name of the computer database run by the NCBI that stores genetic information, that repository of sequence data really didn't exist at all throughout most of my graduate training. So here's a curve that ought to be eye-opening to you. And it shows the growth of sequence data, both in terms of bases here in the blue and number of sequences here, the orange line, from the advent of automated DNA sequencing in the mid-90s until November 2016. Now, that was over 4 years ago. This curve actually now is very much higher. So I need to update the figure to reflect the growth of both of these curves over the past 3.5, 4 years. But you can see this dramatic increase in biological information data that we simply had no clue about back over here. When you have a situation like this in science, where there's a flood of data into a field, it is very likely that the generalizations that you've made over here, sort of out there on the plane, off to the left, where neo-Darwinian theory was formulated, it's very likely that those generalizations will be challenged by this growth of data, and that's exactly what has happened. So here is a common representation of the tree of life in its a rolled up form, because it's hard to represent the tree of life in a horizontal figure, given of course, the enormous diversity of life on this planet. But we see here the 3 major groups: the archaea, single-celled organisms with a number of distinctive features, not present in bacteria, another group of single-celled organisms, both of these lacking nuclei. And then we have the eukarya, and on one of these branches over here is Homo sapiens, of course. We are animals in the metazoa. The key feature of a figure like this that you need to keep in mind for the remainder of this talk, well, there are 2. One is its universality. That is, there's no organism on the planet outside this geometry, and the existence of LUCA, the last universal common ancestor. Now, LUCA turns out to be critically important to how we build the histories of life, or how we infer the history of life on this planet, because LUCA, if it existed, is a biological singularity that strongly constrains what we can infer. I'll explain more about that in just a moment. So if LUCA existed, there are constraints theoretically on what we can reasonably infer about the subsequent history of life. If LUCA did not exist, however, then all bets are off, because how we interpret phenomena such as biological universals, molecular homology, the very rules of phylogenetic inference, everything in historical biology changes if LUCA did not exist. And in the middle third and the last third, so the last 2/3 of this talk, I'll be discussing this in more detail, but keep that in mind that Luca carries a great deal of weight theoretically in how you try to reconstruct the history of life. In particular, evolution is not an unconstrained process. So here's Luca, last universal common ancestor that arises by some unknown abiotic pathway to the living state. And we can reasonably infer that Luca would have had a fairly large genome, probably on the order of about 300 proteins, dozens of large RNAs, and so forth. And I won't go into any detail about this today, but there's a strong case to be made that if Luca was anything like the cells we see today, it would have had a fairly significant armamentarium of different proteins and RNAs for its basic functions. Now, you have to keep what is essential by necessity. So if there's some molecular feature of Luca that's essential for its existence, that feature will be essential for the offspring as well. And as you climb up into the tree, probabilistically, you are going to retain those essential features as you work your way up into the branches of the tree of life. Ergo, you have good reason to think that it's more likely than not that much of this hardware should be present as you work your way up into the branches because of the viability constraints on any evolutionary pathway. Okay, so bear that in mind as we begin to look at orphan genes and at some of the molecular details, particularly in the second half of the presentation. Now, orphan genes have their significance have the great significance they've attained for lots of reasons. The one I'm going to explore today is their relation to the theory of universal common descent, which I've abbreviated here just as common descent. So this is Darwin's tree. Now, it's very difficult to go directly from the proposition all living things on the planet share a common ancestor to observation. So you can check to see whether this is true. Usually what happens, in fact, I would say invariably what happens is common descent is coupled with some auxiliary theory from biology, from some other part of biology that enables us to make predictions that we can then check. So there's a theoretical coupling here that takes this abstract notion of universal common ancestry, links it to something else we know in biology, and then that lets us make predictions that we can check. So to give you an example, for vital functional reasons, the genetic code must be invariant. So strictly for the sake of argument, assume that this is the case. That allows you to predict the universality of the genetic code. Now, what happens when those predictions fail? Something over here in this theoretical coupling, in this bundle, must pay the price, so to speak, for that failed prediction. It could be the auxiliary theory is wrong, or it could be that common descent is wrong, or perhaps both. But something over here has got to pay the price. And the logical puzzle, or the epistemological puzzle raised by orphans is, when we observe them, where is the price paid for the failed prediction that came from common descent plus auxiliary theory? And the problem at the deepest level is, if the price is always paid by the auxiliary theory, common descent is rendered effectively an axiom. It becomes invulnerable to challenge by observation, which I would say is not a healthy state for a science to be in. Even if you're 100% persuaded that common descent is the case, it's still an empirical theory. It's not a proof in geometry, right? It's not a mathematical theorem. It should be vulnerable to challenge by observation should that arise. So this schema of where does the failed prediction end up in terms of the price being paid over here, keep that in mind as we look at orphans. So I actually have a chapter on this coming out later this year in this new anthology coming from the editorship of Bill Dembski, Casey Luskin and Joseph Holden. And the title of my chapter is Can Universal Common Descent Be Tested? So keep your eye out for that. Now, enter the ORFans, which, by the way, are known today mainly as taxonomically restricted genes. So this usage of ORFans tends to be, is tending to die away, and they're known mainly in the literature as TRGs, short for taxonomically restricted genes. Now, the term ORFan itself was coined, I believe, in July 1996 by a French researcher in a review article on the yeast genome. And you can see the heading here, The Mystery of ORFans. In the review article, Dujon reports that approximately half of all the protein coding open reading frames revealed by the yeast sequencing project had no homologs, that is, no matching sequences in any organism, including yeast itself. And this was a great surprise. And he's talking here just about chromosome 3 of the yeast, but he says that's the most striking result. So in the abstract of this review article, he says a significant proportion of yeast genes are ORFans of unpredictable function. Now, functional annotation and status as an ORFan are intimately related, as I'll show you in a moment, via the way that sequence data are deposited in places like GenBank. So ORF is an acronym that stands for Open Reading Frame. It's a sequence of DNA that codes for protein. And by the way, this talk today, I will be addressing only protein coding sequences. I am not going to talk about sequences that code for functional RNAs, of which there's a whole menagerie now of different types. So please keep that in mind. We are talking today only about protein coding sequences. So this cartoon we can take to be representative of a typical prokaryotic open reading frame, where you have a promoter upstream here, a start site for the beginning of transcription, and then ultimately the RNA product will be the beginning of translation. So you have a start codon here, a long sequence of codons with no stops, unless they're specially translated in groups that do that, where an in-frame stop is inserted into the protein as pyrolysine or selenocysteine, but we'll leave that aside for the moment. So no stops, and then a stop signal. And this will be long enough, a long enough sequence with no stops to code for something, right? Although there can be very short proteins as well, but these features will be sufficiently general that you can write algorithms to look for them. And gene finding programs will go through sequence data looking for these features that characterize open reading frames and tag them and say, this is very likely a sequence coding for protein. So you take your newly sequenced DNA, and of course, this is just a cartoon. A real sequence would be very much longer than that. And you submit it to GenBank using some matching algorithm like BLAST, basic local alignment search tool, and you get a hit. So base pair by base pair, you find a high degree of similarity between your new sequence and one that's already deposited and annotated within GenBank. So think of this again on a parallel to natural language, taking a word you've never seen before, going to the English dictionary. And the deposited sequence is annotated as coding for a ribosomal protein. Well, that tells you what your newly sequenced DNA is doing. Now, this genetic word, so to speak, can join the dictionary, and thus the dictionary grows. Except with ORFans, you don't find matches. So the report that you would get from BLAST is no significant similarity found. In other words, you submit the word to the dictionary, and the dictionary, as I showed in that little cartoon earlier, shrugs its shoulders and says, well, I don't know what it does, right? Even though the sequence bears the features determined algorithmically by your gene finding program of very likely coding for a protein, right? It's got a long open reading frame. It's got to start. It's got to stop. It's got the right upstream features. It looks like it should be coding for something. When you submit it to the dictionary, you do not find a match. Hence the pun, orphan, child without parents. Now, when this was noticed, it was noticed very early on. So Russell Doolittle, who spent his career at the University of California working on this and related puzzles, in a review article in Nature in 2002, he said, "This is the biggest surprise. The large number of putative genes with no known function." Again, notice the connection between orphan status and no known function. And that's because when sequence data are deposited, they aren't just dumped into GenBank. Typically, they are annotated with respect to likely function. So absence of function indicates that there's been no corresponding match found in the non-redundant database, NCBI non-redundant database, that would let you assign a function to your new sequence. So he's looking at the signal right about here, okay, when he's writing this review article. Just a few dozen genomes are available at this point. The signal now is very much stronger, of course. But he goes on, how could there be so much in the way of unknown equipment? Where these unique sequences are coming from and what they do remain baffling mysteries. Now, I need to make a distinction that I failed to make a few years ago in a talk in Florida. And in the audience was a correspondent for the blog Panda's Thumb. And he really made me pay for this. And the pain was sufficiently great that I vowed that I would never give a talk on orphan genes again without making this distinction clear. So orphans as a class of biological objects fall within, and this is a Venn diagram, genes of unknown function. But genes of unknown function may have homologs, that is, sequences of high similarity found in other species. And orphans, by their strictest definition, are found in a single species. So the fact that a gene of unknown function has a match in some other species means it can't be an orphan. So this class is larger than this one. The mnemonic is all orphans are genes of unknown function when they are first discovered, but the opposite is not the case. So there, you benefited from my pain at failing to make this distinction and paying for it. Richard Buggs and I collaborated on a chapter for Cambridge University Press that was published in 2016, where we discussed at the time, now it's 4 years later, so the puzzle is much more interesting today. We recounted the discovery of orphans and their significance, and the editors to this volume said very nicely in their summary, every genome study to date has revealed an unexpected percentage of unique genes. An increased sampling of gene space has resulted in this space expanding rather than contracting. So the point they're making there is if this were simply a sampling artifact, one would expect that as more sequences came in, the orphans would go away. And that, in fact, has not happened. And they point out many of these appear to be functional. So the reason that Richard became interested in orphans is about the same time our chapter came out, he and a group of collaborators published the genome of the ash tree. And so Richard, if you know him, he works on diseases of plants, in particular diseases of trees. And ash dieback is a terrible disease that kills ash trees in the EU and the UK. But some subspecies of ash appear to be resistant to this disease. So Richard was looking in a variety of ash genomes to try to find the molecular correlates for resistance, right? That would be very, very useful to people trying to save the ash tree, that genus. Well, when they sequenced the ash, they found that approximately 25% of its protein coding genes were orphans. So Richard wrote up a commentary for Nature, Ecology, and Evolution talking about this, calling it an evolutionary mystery. And he's continued to maintain his interest in this puzzle right up to the present. But let's go back to our library briefly. You point out to me, Paul, every book I look at has words that I haven't seen before. All right, how does this work? How are orphans defined when we pull these books off the shelf? So I'm going to jump between my analogy and biology. What is the actual analytical pipeline by which we decide that a gene is an orphan? Well, here's our new sequence data. Okay, so we've opened another book. And we have a taxonomic category, species, typically species, sometimes genus. And we take our sequence data from this species and we compare it to what's present in the non-redundant database, the NCBI database of all known gene and protein sequences. And we set a homology threshold. Now, this is a proxy for yes, the same, no, not the same. Okay, and in BLAST, it's an adjustable parameter. It's called the expect value. And you can change the expect value. You can make it more or less stringent. But a typical expect value for assigning orphan status is 10 to the minus 3 to 10 to the minus 5, although people can go those negative exponents can get very much larger depending on how stringent you want to be. And through that pipeline, you submit your DNA sequence data, and what gets kicked out down here are the protein coding sequences found only in one species or genus. So that's how you're isolating these unique words. Now, when this was discovered, again, this is following on what Russell Doolittle said, people were scratching their heads because it was unexpected. This finding was unexpected. The kind of thing that happens in science that makes science so fun and interesting when nature talks back to you. So this Israeli team said, each of these orphans is a mystery awaiting interpretation. How could they be sitting out there in sequence space with no kin, right? Well, why the surprise? Now, here we need to put on a philosophical pair of glasses, if you will, and say, well, what's in the background theoretically against which orphans are so surprising? Well, Doolittle wrote another review article now in 2005, looking at a stronger signal from the whole genomes that were available. And he said, we thought we could identify extant genes in silico because in the theoretical background, we had the expectation all extant genes descended from a smaller ancestral population that expanded by gene duplication. So in this cartoon, again, we have a DNA sequence here coding for a protein that folds up like this. That sequence is subject to a variety of evolutionary processes. And let's say there's an internal duplication and we add an alpha helix here. Well, these would still nest together at the protein data bank because their overall fold is going to be the same. This one has an additional structure here, but these are going to be classed in the same family of proteins. And these DNA sequences, they will still be a trace of history because these evolutionary processes will not totally scramble the information that's present here in the mother sequence. The daughter sequence, you will still be able to identify via BLAST or some other matching algorithm, such that if you pick any protein or its gene up here, let's say this is Pantroglodytes, the chimp, and this is not a proper phylogenetic diagram, by the way, it's just a cartoon, and you work your way back down its lineage, you ought to be able to find its molecular kin as you head back down towards Luca. So this is the geometry that Israeli team and Doolittle and really pretty much everybody else in the field had in mind when orphans came along. So what's surprising them is common descent 1 is a given. It's not up for grabs. We think we know how new genes and proteins arise, and that process will leave a trace of similarity, more precisely of homology. If 1 is the case and 2 is the case, the last thing one would expect to find are a lot of unknown genes and proteins. Now, you need to keep in mind just how deeply surprising orphans were, because until their discovery, the origin of orphans from random DNA was seen as all but impossible. So I lived through this as a student. It was textbook orthodoxy that the origin of a novel gene with a functional protein product from otherwise random DNA was effectively zero. And you find this all throughout the orphans literature. Often people will open their papers by saying, nobody expected orphans, and here's why. So in this 2009 paper, the author says, it was looking to be like transmuting lead into gold, right? You would not be able to get functional genes with all their features directly from junk DNA for a variety of reasons, right? The origin of novel functional proteins from random DNA would have been, prior to orphans' discovery, viewed as effectively impossible. Part of the reason for that is to have a functional open reading frame. There are non-trivial requirements on that sequence, such as having a promoter in the right position, being able to have its protein product be incorporated into the cellular context and do something, right? Have a role to play. And the cellular context cannot be ignored. Cells, all cells that we know, do not like broken, unemployed, or otherwise homeless proteins. They tend to be very bad actors. They aggregate, create all kinds of mischief. So there's a whole system dedicated to cleaning up the protein junk. Ubiquitin tags are attached to homeless, unemployed, or broken proteins, and they are dumped into the proteasome where they're chewed up. This is the cellular garbage disposal, and this system is essential in eukaryotes, certainly, and apparently also in archaea. All right. Now, what happened when orphans were discovered? Well, the wind changed direction 180 degrees. So this is Aoife McClysik. I hope I pronounced her name correctly. It's Irish names are not something I'm terribly comfortable with, and I apologize to her if I mispronounced it. But she's one of the leaders in this field. And in this paper in 2015, she said, look, we just didn't expect them. De novo origin was usually considered so improbable as to be impossible for more recent evolution. But here the orphans are, right? In yeast, plants, flies, mammals, and even in humans. So what happened was the wind changed direction. Apparently, de novo origin happens all the time. Now, again, we have to pay attention to the deeper logic. If we have an auxiliary theory, as we did until orphans were discovered, that said there's a very low probability of the formation of proteins de novo, that predicts few, if any, orphan genes and proteins. What happens when orphans are discovered? Because something over here went wrong. Okay? Now, what's tended to be the practice of most biologists working in this area is to say we were wrong about this. Our auxiliary theory was wrong. However, some biologists have begun to say maybe the problem is here. And we'll look at some in a few minutes. All right. I'm on schedule. I think I will finish on time, which is going to be great. So whole genome sequencing has enabled all kinds of experimental approaches that were just not available throughout most of the 20th century. For instance, the use of transposon mutagenesis to determine what are the essential genes and proteins in any particular species. So these are not to scale. I've just made this big so you can read the name. Transposon is a piece of DNA that will locate itself randomly in other DNA. So let's say we introduce this transposon into a bacterial population, and here's a circular bacterial chromosome, and we have 4 integration sites, and at least one of them codes for a protein that's essential. So the transposon disrupts that open reading frame. The cell says, you know what, I couldn't do without that protein, and the plates are sterile, right? We come in Monday morning, there's no growth, the cells are dead. Same thing here. At least one of these integration sites hit essential hardware, no growth. Sometimes the transposons will integrate and we'll have colonies on our plates, right? The cells are alive. Maybe they're not doing terribly well, but they're definitely alive. So we do this often enough, and we look to see where the transposon integration sites are in the bacterial chromosome. And by being thorough, we can work our way right around this chromosome. The fact that we see a transposon in an otherwise viable cell tells us that at least under laboratory conditions, whatever was at that position, the DNA there and its protein product were not essential. Whatever this experiment is done, the results are fascinating. It's been done many times. I'll report here on just one of these, done in Lucy Shapiro's lab at Stanford. They used Colobacter, which is a bacterial species that you can culture out of fresh water. So they did this transposon mutagenesis experiment with Colobacter, and they found a little over 1,000 essential elements, including 90 segments of unknown function. Remember that connection between unknown function and orphan status. So these are orphans. Nearly a tenth of its essential hardware for Colobacter is orphans. So in an interview, when the paper was published, Shapiro said, "We don't know what they're doing. We just know the cell can't live without them." All right. But here's the real moral of this experiment for evolutionary relationships. Colobacter and E. coli are in the proteobacteria. So they're in the same large bacterial clade. When you look at this Venn diagram, and this is a picture from a figure from the paper from Shapiro's lab, Colobacter and E. coli are not running their basic biochemistry or molecular biology using the same hardware. There are significant fractions of their genomes that are species specific. What that tells you, and this is a very homespun analogy, imagine no one in the UK would have the horrible taste to do this. So this is definitely an American auto show, all right? You work your way down this row of engines and you notice that the essential parts are some shared, some not shared. All right. The essential parts of these engines are not the same. There's been significant tinkering with them in terms of the parts that they can't do without. Again, this pattern of what's called now non-orthologous gene displacement, that's a really horrible long name, but the phenomenon is quite general. And as I said, it's seen right across different groups where transposon mutagenesis is used to identify essential hardware. So some of you doubtless remember this paper, a really amazing experiment from Craig Venter and colleagues constructing a synthetic bacterial chromosome, SYN 3.0. There it is. 1/3 of its 473 essential genes were unknown function. You know, arguably close to the simplest free living thing we know, very carefully engineered by Venter and his group. I mean, in a technical tour de force of building this bacterial chromosome over 1 million base pairs, a lot of the hardware, they didn't know why it was there. Those are orphans, at least they were at the time. Okay. Here's another example. My phone, if I turned this screen, you would see my office phone sitting over there. The cord does this all the time. It super coils on itself. It's very annoying. I have to unplug the cord from the handset, let out the supercoiling to make it functional. Well, the geometry of DNA faces the same topological problem. Topoisomerases are essential proteins in all living things we know to handle the topological difficulties that arise with a spiraling double helix molecule like DNA. So topo5, this protein is found, as far as I know, in a single species. It's an archaean, lives at temperatures that would be very uncomfortable for you and me. But this topo turns out actually to be commercially valuable because it is stable at high temperatures. So it's used in lab kits to process DNA. And I love the title of this paper by Forterre in France, A New Fold of Mysterious Origin. In the paper, he says how mean of Mother Nature to give this topoisomerase to a single species. But he's alerted to the unusual fact that this essential protein is found in a single species. So he and a colleague go out and look at the distribution of topoisomerases throughout the tree of life. Now we can do a thought experiment here. What would be the case if this geometry were true? So this is another representation of the tree of life here, kind of viewed top down. Leuca is here. So we have the archaea, the bacteria, and the eukarya, all sharing an origin at Leuca. We would expect one ubiquitous topo 1 and one ubiquitous topo 2. Topo 1s, very nice terminology here, they make a cut in one of the sugar phosphate backbones. Topo 2s cut them both. Topo 1s and topo 2s are essential, or some player that does that job in all living things that we know. Our expectation would be that they should be ubiquitous throughout the tree of life. In fact, that's not the case. So Forterre says one would naively expect to find two homologous topoisomerases descended from those present in Leuca. An intelligent designer would probably have invented only one topo 1 and one topo 2. Reality has turned out to be quite different and more interesting. The actual distribution does not or is not congruent with the universal tree based on 16S ribosomal RNA. So here's a figure from their paper, and you can make out the three major domains. Archaea here divided into the three groups, eukaryotes, bacteria. There are so many different topoisomerases and other DNA disentanglers, so to speak, that they hypothesize ancient viral reservoirs to give rise to them. But there's really no phylogenetic logic here that corresponds with the three-domain picture. Not only that, in other words, this lack of congruence between the distribution of essential proteins and the three-domain geometry, orphans are not going away. So this is a very early survey. This is 16 years ago. A group in the UK looked at what would be the trend line for the discovery of orphans. And they said, look, it's continuing to rise in a roughly linear fashion, despite the growing number of genomes sequenced, and it shows no signs of leveling off. So here's a figure from their paper, and they've binned their data into two groups. This group here is are the orphan genes longer than 150 amino acids. Most orphans, or many of them in any case, are shorter than that. So this line here, these are those shorter than 150 amino acids. And you see there's a big jump here where this species had a lot of orphans. Now, I presented this figure at a talk at Dartmouth University shortly after this was published. And there was a microbiologist in the audience sitting down in the front row. And he was very unhappy, visibly unhappy. So the Q&A arrived, and he shot his hand up, and he said, Paul, what's in the numerator position? So their study was based on 122 microbial genomes. So I said 122. He says, what's in the denominator? I said, I don't know. And he said, nobody knows, but it's bound to be this, right? You have a terrible sample. You have a tiny fraction of what's out there in the biosphere. You're doing what should never be done in statistics, which is generalizing over a crappy sample. So at the time, in 2005, one of the standing objections to the reality of orphans was they were a statistical artifact of a bad sample. If I ever get back to Dartmouth and he's still on the faculty there, I want to look him up because he was wrong. I mean, if the objection was reasonable at the time, it just wasn't true. 4 years later, a group at the University of Connecticut did a very interesting experiment. They sampled from 573 bacterial genomes randomly, picking genes one at a time and saying, okay, who else in the 572 genomes has this gene? And what they found was the distribution of the genes was very striking. So they have two ways of representing their data here, as a pie chart and as these absolute number balls here. So the pie chart is representative of a typical bacterial genome, where you have 8% sequences that are universally shared. They call that the extended core. 64%, which they call the character genes, that are in a smaller, they have a non-universal, smaller distribution, but still taking in a larger section of bacterial space, so to speak. And then 28% in the pie chart, unique to that group. Well, if you look at the way you count these, you've seen these before. So their absolute number is going to rise relatively slowly compared to this one. Because each of these is unique, their absolute number is going to rise much more rapidly. And they said in their paper, the bacterial pangenome, that's the genes of all bacteria within all cells within the bacterial domain, is of infinite size, which is hyperbole. There are not an infinite number of bacterial cells on this planet. There are a lot of them, but it's not infinite. So really, this should be indefinitely large, meaning you'll never see the outer boundary. And it's essentially a counting problem. Because you've seen these before, and they tend to be ribosomal. You've seen one, you've seen it before, its count will stay relatively low. This will grow somewhat more rapidly. The ORFans, by contrast, will be enormous. They will swamp the total number of shared sequences. This German group in 2011 said the total number of ORFans across all lineages by far exceeds the number of known gene families. And there's no reason to expect this to change. This sampling curve is not going to flatten. So this ORFan anomaly and others have given rise to a great deal of disenchantment with a single tree picture. So I'm giving you just a sample of expressions of opinion in the literature by people who have no interest in intelligent design really whatsoever, but they can't fit their data to the single tree geometry. So Raoult in Marseille, who became notorious over the past year for advocating hydroxychloroquine, I think it was, as a therapy for COVID-19 infection. In 2012, with I think a postdoc, he said, tree of life concept, it's not true. It's contrary to current knowledge because of ORFans. For Doolittle, who's been sort of the grand old man of this puzzle, in his inaugural paper for the Proceedings in the National Academy, when he was elected as a foreign member, he said, Darwin said we had a tree of life. It was a fact of nature. But look, there's no independent evidence that living things fall into a single tree. And the tree of life rests on unproven assumptions that are unlikely to be broadly true. Eugene Koonin at the NCBI, in a paper in 2007, he said the tree of life does not exist. And Craig Venter, Mr. Human Genome himself, said, I don't buy it. Counterintuitive. We may have thousands of recent common ancestors, and they are not necessarily so common. So one of the phenomena that leads Koonin to doubt the single tree of life picture is what I referred to earlier as non-orthologous gene displacement. Hideous, kludgy name for a very interesting puzzle. So let's say in species A or species 1, we have some pathway enabling function X where genes A, B, and C are demonstrably essential. And then we look at function X in species 2, and we find instead of A, K, B is still there, but instead of C, Q, but they enable the same function. So non-orthologous means that these genes, when we compare K to A, they exhibit no molecular similarity, or it's beyond the threshold of what we would consider orthology or homology. Same here for Q and C. Yet they're conveying the same function. So one of the things that led Koonin to this conclusion is his study of the DNA replication apparatus in different groups where he said it looks like DNA replication, the proteins involved in this process arose twice independently because of the lack of homology between that machinery in bacteria and the machinery conveying the same function in archaea and eukarya, but the proteins are not homologous. The genes are not homologous. So more recently, in 2016, he said, look, the universal core of life has shrunk almost to the point of vanishing. In fact, there is no universal genetic core of life, owing to the near ubiquity of NOGD. There are a few still apparently universal proteins. They tend to be ribosomal, but every new genome that comes in pushes the universality number down. So Kunin no longer accepts the tree of life. So I'm going to have to move rapidly through the animal examples to finish on time. I apologize for that. And if anyone contacts the leaders of the UK ID group, I will make available to them the PowerPoint. And my only request, if you want the PowerPoint, is do not put it up on the web. Use it just for your study. So this lab in Germany has taken animal orphans very seriously. They had a beautiful paper looking at the anatomy of Hydra, which is apparently specified by a novel species-specific protein family found only in Hydra. This is a standard phylogeny for the phylum Mollusca, showing the major groups here with a common ancestor at this node. One would expect, if this geometry were the case, that the proteins, the genes and proteins involved in the mantle tissue that gives rise to the calcareous structures present in these different groups would be homologous because they all inherited it from their common ancestor back here. In fact, this group in Australia found that these groups are building their mantles and their calcareous hard parts using species-specific secretomes, lineage-specific. So I apologize for this, but I'll explain what's going on here. This is a heat map showing at the purple end, a high number of species-specific sequences, and then going down the other direction towards zero. If this geometry were true, one would expect the dark colors to be here because they're being inherited from a common ancestor, and colors getting lighter as we move out to the terminal branches. In fact, just the opposite is observed. At the terminal branches where the species are is where the species-specific important secretome proteins are. Exactly the reverse of what one would expect on a hypothesis of common ancestry. I'm going to skip a few slides just to stay on time. In 2010, 2011, 6 ant genomes were sequenced, and on initial pass, they had very high fractions of orphan genes. Well, you have to compare these ant genomes one to another. When that's done, the orphan fractions come down. So the revised figure here for the fire ant is 5.6 orphans, 11.1 and 10.3 for these two species respectively. In these cases, the revised numbers came down to 8.9, 12.6, and 11.4 orphans, respectively. Now, we are effectively doing a random draw from 14,000 ant species worldwide. These have been chosen for their biological interest, like leaf cutters are incredibly interesting, or their agricultural pests. But we're effectively picking at random from this very much larger group of 14,000 ant species, 6 to sequence. There's no reason to think we just happened to pick the 6 that had anomalously high orphans fractions. So we can take an average from these 6, which comes out to about 10% orphans per genome. And we can run the numbers. Just for the Myrmecinae, a very large group clade of ants, 5,800 species, average genome size of about 17,000 protein coding genes times a 10% orphans fraction gives nearly 10 million unique genes just within that group. What happens when we consider all 14,000 ant species using the same calculation? Well, here's the probable genome size of the common ancestor of ants if it existed. The number of orphans swamps what's shared. If this box is about 12 feet on a side, this box is 260 feet on a side. Of course, these figures are not to scale. And orphans are omnipresent. When I presented a talk on orphans, probably, I think it was about 13 years ago, in a meeting in Chicago, Francis Collins, who's the head of the NIH and a geneticist, was there. It was a small meeting about 20 people. And Collins was sitting very close to where I was standing giving my talk. And when I finished, he went like this. He was very kind, wasn't hostile, but he was unimpressed, let's say. And he said, Paul, nobody cares. He said, orphans are found in viruses and prokaryotes, right? Archaea and bacteria. But until we find them in abundance in animals, no one's going to care. Well, they are in animals in abundance. This little tick, this number may have changed now, but on first pass, over 2/3 of its protein coding sequences had no sequence homology to the proteins in this database. In the red flower beetle, Tribolium, it builds its legs using an orphan, the flip-flop protein, okay? Not found in any other insect genome. This very cute little bug, a leafhopper, produces a superhydrophobic coating in its Malpighian tubules that it deposits on the outside of its body. And these are the brachysomins, the proteins involved. They look like little buckyballs. They're unique, not found. The proteins involved in making these are not found anywhere else. This, I think, is the most dramatic example. It comes from a group at the University of Toronto. They looked at the putative last common ancestor of the domain Eukarya. By sampling from different eukaryotic groups, looking at the genes and proteins involved in chromosomal segregation. So here we see meiosis. This would occur in any sexually reproducing eukaryote where you have chromosome reduction by meiotic division. And they looked at the machinery involved in this process of pulling the chromosomes apart. It turns out that in very well-studied groups like C. elegans and yeast, the genes and their protein products involved in this essential eukaryotic function are orphans. So in their paper, they said, why should so many taxonomically restricted genes play key roles in this core process? Well, maybe the reason is there's more than one operating system for life. And the expectation based on universal common descent is just not true. So last slide. Luca, as I told you at the beginning, creates theoretical expectations for us. If Luca existed, we can project a radius from it that is defined by three things: the functional demands of viability, there's no dying in evolution, known mutational processes, and time. If we can define these with rigor, we can project a radius, right, over 3.8 billion years that tells us we should not expect to observe genes and proteins outside this sphere. There just hasn't been enough time, and evolution is not unconstrained. It's not an unconstrained process. And then we just sequence DNA wherever we can. And we ask, where are genes and proteins falling? Are they falling within this sphere consistent with universal common descent? Or in fact, is that radius very much smaller? I couldn't make this any smaller in PowerPoint. It should be really a tiny dot here than what's available to a designer. So I ran over by 5 minutes, and I apologize for that. But thank you for being so attentive. And I will try to answer your questions. If I can't answer them, I'll try to point you to relevant literature that will help. And again, these slides are available for your use contact the organizers. And again, do not put the slides on the web. Keep them on your own hard drive. Thank you.
Paul, can I start by saying a big thank you for that talk? That was hugely interesting, fascinating. So big thank you. I know how much time it must take to put talks like that together and PowerPoints like that. So thank you ever so much for taking the time to talk to us this evening. Can I start with some questions that have come in on the live feed? The first question is this: if DNA replication is argued to have taken place twice independently, then as more data comes along, might it not be that it evolved more than twice, possibly a huge number of times, independently?
Yes. One of the things that's happened within evolutionary theory that's really surprised me, especially over the past decade, is the revival of theories of polyphyletic evolution. So I omitted this slide from the talk because I was worried about running over, but you can divide the landscape of opinion on this question into 4 quadrants, one of which is no tree of life, but also no design. In the 19th century, Darwin was very much the exception among evolutionary thinkers for arguing for a single origin. Most evolutionists had a polyphyletic view. Ernst Haeckel, for instance, thought that spontaneous generation was happening all the time. So the single-celled organisms we observe today arose in recent history. They were not the lineal descendants of Monera, as he called them, from the early history of the planet. So there is an increasingly large school of thought within evolutionary theory that these events may have happened multiple times independently, given the right starting conditions. So the questioner asks exactly the right thing, which is, well, look, if it can happen once and more than once, maybe it can happen n times, where n is to be determined by looking at these systems and saying, what are their constraints? How much can they vary? So yes, I think that's entirely possible. And I've been surprised, as I said, by the growth of that school of thought within evolutionary theory.
Okay, thank you. Next question is this. When genomes are sequenced, like the ants that are compared, what proportion of their genomes are actually sequenced? Is it 100%?
Well, sequencing is not the hard part. The hard part is constructing the genome from the sequence. So in human beings, for instance, we have very large sections of repetitive DNA where the same sequence will occur in multiple copies, thousands or tens of thousands of times along the chromosome. And you can sequence that, but your sequence is going to be fragments of that. And then you have to align those fragments, find their regions of overlap to figure out where you are on the chromosome. So one of the problems of getting a complete sequence for any species, humans or ants, really the only exceptions to this are bacteria and archaea, which have very streamlined genomes, is you can run into one of these sections of highly repetitive DNA and you just don't know how to do your assembly because the repetition doesn't give you a hook or a place where you can begin to align the sequence and reliably construct out from there. So bottom line is getting a complete genome is not really a problem of sequencing as much as it is assembly from the fragments that you have. And I think when you go to a genome browser and look at any species, there will be data there telling you the degree of completeness of that genome. I hope that's answering what the questioner asked.
Okay, thank you. Can I move on? Question on the tree of life or universal common descent. If that concept or idea is torpedoed, as you suggest, how do those who still support common descent seek to sustain their case?
Well, if you think back to the diagram with the common descent making predictions, anytime you have a failed prediction in science, you are generating that prediction from a complex bundle of assumptions, theories, data, and so forth. And in engineering, they call this the problem of failure analysis or failure localization. You need to figure out where in that complex bundle the problem is. Now, it may be in multiple locations, but typically a defender of common descent will say, our auxiliary theories are imperfect, and they will make the auxiliary theory pay the price for the failed prediction. So they protect common descent. The price is paid by suspending the status of the auxiliary theory. It's cast into doubt. Now, you can do that. And in fact, there's a whole body of thought in philosophy of science coming mainly from the Imre Lakatos, that all sciences protect their core theories and make the cost paid by the belt around the core. The problem then is common descent becomes effectively an axiom, invulnerable to challenge from observation. And I don't think that's a healthy position for any science to be in, because we want, to use a different metaphor, we want traffic in both directions on that highway, from theory to data, from data back to theory. Actually, in the chapter I have forthcoming later this year, I talk about this problem, that common descent should not be insulated from challenge. And most biologists I know, frankly, in evolutionary biology, when we're in conversation and I point this out to them, they say, you know what, Paul, you're right. I don't want to protect common descent. Here's what I would take as a challenge if I observed it. So part of why I'm involved in this is I wrote my PhD on the theory of common descent, and I want to see it treated the way every other scientific theory is treated. Give it the honor of failing if it needs to fail.
A follow-up question on that is, how important is it to the theory, or to Darwinian evolution, or neo-Darwinian evolution? And you mentioned some scientists in your talk who appear to have rejected or accept that common descent is no longer sustainable. But unless I'm mistaken, those scientists are still supporters of Darwinian evolution. So to what extent, if any, is common descent fundamental to Darwinian evolution?
Well, if you define Darwinian evolution strictly as the universal tree plus natural selection, then it would be critical to the truth of that theory. But I take evolution to be a much broader body of thought. Certainly from Darwin to the present, there have been many evolutionary theorists, not intelligent design advocates, who accepted a polyphyletic or multiple starting point view for the history of life. In fact, I've got a book right up here by a great Russian zoologist who argued that if life could start once, it could start more than once. And if you raise the probability of the origin of life, move it towards one, you could have multiple independent starting points that evolved naturally. And that's not an intelligent design position. That is a view of evolution, albeit not with a single tree. So I try to stress to my students, don't assume a polarity between naturalistic evolution with a single tree and intelligent design as if those were the only two options on the table. In fact, we have at least four positions. One of my dearest colleagues, Mike Behe, is a design theorist. You guys have had him on, right? He accepts the single tree picture. Alfred Russel Wallace, Darwin's co-discoverer of natural selection, towards the end of his career was a strong advocate for intelligent design. And as far as we can judge, Wallace accepted the single tree picture. So again, bottom line, there are a range of options, and evolution as a general picture allows for more than the single tree. Historically, that's been the case, and that's certainly the case today. Eugene Koonin does not like intelligent design. I can tell you that for sure.
Okay, moving on. Next question. Are orphans included in phylogenetic studies? With 30% or even 10% of orphans, surely cladistic methodologies are flawed.
It's a real problem, especially because it looks like orphans are critical to defining what makes a species a species. So I showed you Tribolium, the red flower beetle. That flip-flop protein that's experimentally been shown to be essential for the development of its legs, that is essential for defining that species. And as you move along the terminals in any cladogram, you find orphan genes that convey particular species unique functions and structures, such that you couldn't have the species without the orphans present in its genome. Cladistics works by shared characters. So all mammals, have 3 ear ossicles, are warm-blooded, mammary glands, and so forth. There are characters that define the clade mammalia. Orphans, by definition, are unique, so they drop out of phylogenetic analysis, except when you get out to the very tips. One slide I skipped over, and I apologize for that, shows a very interesting distribution of orphans where at the level of genus, you often get a big spike in orphans, which is surprising because one would expect, if common descent were true, that you would have most of the genes shared universally and then a steady tailing down, and the least number of genes should be at the most, at the furthest out point, the twigs on the tree. In fact, you get this kind of funny U-shaped pattern where at the genus you get a big spike in unique species, in unique sequences, orphans. So cladistics handles orphans very poorly, I think, by the very methodology.
Okay. The next question starts with a compliment, so I better read that to you as well. The compliment is fantastic talk. And then the question is, if orphans are so common, and if you have many starts of life, might the biological predestination of Dean Kenyon be back in fashion for those common proteins?
That is a challenging question because when Kenyon wrote that book, he was convinced that there was a nearly deterministic or deterministic pathway to the building blocks of life. It's right there in the title of the book, right? That basic biochemistry of life on this planet was, as it were, built in to chemistry itself. And between the publication of that book and the Dean Kenyon of today, he went through a total change of heart about that thesis, mainly because the chemistry didn't work out that way. There did not appear to be a deterministic pathway to proteins, let's say. My own view, insofar as I've been able to develop it, is one of modularity. If you think about natural language, so here's the dictionary I use on a, can you guys see me, by the way?
Yep.
Okay, it's my Random House. This is my working dictionary day to day. I've got the OED up there, but that's for the tough cases. Think of, the words in here are modules. I can rearrange them in a variety of ways. I can write a book chapter on orphans, or I can write a love letter to my wife, or I can write an automobile manual. These lower level modules can be rearranged in various ways. And it's the higher level context that determines their function. So I actually did this exercise a few years ago. I took the last 44 words of the Gettysburg Address, Lincoln, this wonderful, moving address to the American public about the terrible tragedy of Gettysburg. I took those words and I rearranged them, the same words, into an anarchist's manifesto. 180 degrees different in meaning, same words, but the higher level context, the paragraph in which they occur, changes how they function. So when I see homology between, let's say, a DNA binding protein or a cytochrome or some other basic module, I say that plays a particular role within a larger system. And I see no reason that a designer should have to reinvent things every time he moves to a different system, given that the system as a whole determines how its parts function top down. So I don't think chemistry does lead to life. I think chemistry tells us if you want life, you're going to have to build it. It's not going to arise bottom up deterministically. So that I'm sorry, I'm wandering a bit, but I would not take the biochemical predestination thesis of Kenyon and import it into an intelligent design framework. And Kenyon wouldn't either, right? He disavowed that book.
Okay.
I think it was in part writing that book that led him to disavow it.
Okay, next question. Regarding the cost of sequencing and computational power, where are we at? What's the next big milestone that would make this more cost effective and advance our understanding?
Well, there is a technology that I want to get for myself that is nanopore sequencing. You can buy from Oxford Nanopore, I think that's the name of the company, a min-ion sequencer that's about the size of this, a little bit bigger than this mouse, right? And plug it into your laptop with a USB cable. And the prep is desktop. I mean, preparing the DNA for analysis can be done by a high school student with sufficient training. I think the last time I looked, the min-ion, the individual sequencer was about $1,000. I don't know that you could plot this along sort of Moore's law kind of path, but cost is coming down, 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 down. And I fully expect that within our lifetimes, high school biology classes in the UK, here in the States, elsewhere, those students will be doing sequencing in their classrooms of species that no one has sequenced before, because the technology will be so accessible and the cost will be so low that it will be realistic. In fact, it may be happening already for a high school advanced biology class to do their own DNA sequencing. And as I said, it's not the sequencing that's the problem. The real challenge is the assembly. From the sequences you get, building back to the complete genome. But that, I think, is also being improved every year. The algorithms get better. It's really the golden age for biology. Physics is in a bit of a muddle, but biology, we have more data than we could possibly handle, and it's pouring in at an increasing rate. So I tell my students, you want to fund science, go into biology.
Okay, I've got two more questions on the technical side, if I can put it that way, and then two moving on to the wider question of intelligent design and the intelligent design movement. First of the technical is this. You mentioned that most orphan proteins are less than 150 amino acids. Is this shorter than most functional proteins? If so, do you think there is significance to this, e.g. in the role of these proteins?
Well, as I indicated, their shortness is striking, but many of them are what are known as disordered proteins. In other words, they don't fold up into they don't on their own fold up into stable structures that allow you to determine their structure crystallographically. It looks like they associate in large complexes. So their length isn't what's really relevant. And their sort of intrinsic disorder is an indicator that these are smaller parts of much larger conglomerations. And the real acid test for orphan functionality is something like targeted deletion. So shorter length is interesting, but I don't think it's really the point analytically where you would say, what's the biological importance? If you can target a deletion for a gene, for an orphan protein, and you have a clear phenotype from that, I would say that's a good signal that that orphan is functional. And typically when functional assays are done on orphans, not only do they turn out to be functional, they turn out to be very important for their respective species. Interestingly, the Berkeley Structural Genomics Group set out to solve the orphans in the genus Mycoplasma, sort of one after another in terms of their crystal structure. And many of them, I think over a third of the orphans in Mycoplasma exhibited novel folds. So I really do think they are a gold mine for discovery. And I think the question to that was an excellent question.
Okay. And the last technical question is this. Is it true that TRG are short? And if so, why?
Many are short. I think this question overlaps with the previous one. I think that many of the longer proteins were already characterized, and that TRGs have products whose short length is associated with their functional role in species-specific complexes. So intrinsically, so-called intrinsically disordered proteins are only intrinsically disordered when they're considered on their own. But in complex, when they associate with other proteins, they form stable, larger scale structures that appear to be very important. So again, I would urge the questioner, don't focus on the orphan in isolation. Look at what may be its causal role or functional role within a larger system.
Okay, a slightly wider question that's just come in is this. Again, compliment is a great talk. And then it says, is common underlying biochemical machinery of life not enough to infer common descent, even with widespread occurrence of unique genes and proteins?
I would say there's a missing premise there. And the missing premise has to do with the probability. So we need to think, are we thinking as design theorists, or are we thinking as philosophical naturalists, where we have a limited toolkit? So we're setting aside the hypothesis of intelligent causation, assuming arguendo that physical process will provide our toolkit for explanation. The missing premise in the question is, what is the probability antecedently of the origin of any biological system? So typically, for something like the origin of the genetic code or the origin of the ribosome, the investigator assumes that the structure in question could only have arisen once. Francis Crick famously talked about the genetic code as a frozen accident. Well, you don't freeze the same accident twice, right? So Crick's mature view was the genetic code has the features it does because once it got up and running, by whatever pathway that is, by the frozen accident of it actually working, it was fixed in place. And so when he was writing, the genetic code was widely assumed to be universal. Now we know that's not the case. The NCBI maintains a webpage with dozens of variant codes. But let's assume Crick is right. The accident is frozen once, it's universal. That pathway for Crick had a very, very small probability from the prebiotic state to the code. But what happens if we move that probability away from zero, where it was for, or near zero, where it was for Crick or Dawkins or Ernst Mayr, take that probability and scoot it on the interval over towards one, and say, you know what, it looks like an accident, but if we could work out the full pathway, we would see that it was bound to happen given the right antecedent conditions. In a situation like that, having biological universals like DNA, RNA, protein, that system is present in every cell that we know. Having biological universals may not indicate a historical singularity, but rather you want to be a cell on a planet like Earth, right? Given this chemical starting point, you're going to have a ribosome, you're going to have an information polymer, you're going to have, X, Y, and Z. So this view was held by Christian de Duve, for instance, the Nobel laureate I met at 20 years ago at Baylor University. De Duve's view was assume that the origin of life is overwhelmingly likely to occur, right? That it's not a one-off event. It's bound to happen. And then multiple independent starting points becomes not only reasonable, but maybe even the best hypothesis. So I resist the move to infer a singularity, thinking now strictly as a philosophical naturalist, because I don't know what the probability of those pathways would be coming up from chemistry towards the living state.
Okay. Now, can I move on to two questions about the wider question of intelligent design and what might be said to be the intelligent design movement? And you, of course, have been linked with that movement for many years. The first question is this. There's come in, it says, how can people that live in the Middle East contribute and help intelligent design?
Well, get in touch with me. The organizers of this seminar have my email address. I have been working in this area for almost 40 years. I'm getting pretty old. I have an international network that I'm affiliated with, and other people are actually on this particular seminar are involved with, that has representatives in Turkey, Israel, Italy, the Far East, Brazil, all throughout the EU, Russia, Japan. In other words, there is a international community of people interested in intelligent design that's growing all the time, and some very good scientists, very good philosophers in that community. So get in touch with me and I can slot you into that network. It's wonderful and exciting. So I'm just an email away.
Okay, and the last question tonight, Paul, is this, and I'll read it out. It's coming to me by email. It's a couple of paragraphs, and it's this. Intelligent design is said to be non-religious. So my question is this. Why refer to an intelligent designer and not intelligent designers? As you say, or it's said, the evidence can't speak to the characteristics and nature of the designer, and therefore can't say one way or another whether it's one designer or more than one. It seems to me that by referring to a designer in the singular, it's closing off, either intentionally or unintentionally, the prospect of there being more than one designer. And in doing so, it's actually reinforcing religious connotations, at least so far as those religions who consider there to be one creator go. I've never heard a proponent of ID refer to the possibility of there being more than one designer. So, to repeat, the attempt to neutralize religious connotations when looked at objectively is actually reinforcing the religious idea of there being a single creator. How would you respond to that?
Well, interestingly, this year, a Harvard astronomer, Avi, I believe his last name is pronounced Loeb. He's Israeli, but he's been at Harvard University for many years. Just published a book arguing that we have the first decent evidence for extraterrestrial intelligence in an astronomical anomaly, Oumuamua. It's a Hawaiian word for scout, I believe, that in, I think it was 2017, passed through our solar system at a very high velocity, describing a path that does not fit with normal cometary motion. Bottom line, this Harvard astronomer says he has a book out about this. It's been widely reviewed, and you can listen to podcasts with him. It's absolutely fascinating. I think the nature of intelligent design is not tied necessarily to a single designer. By that, I mean the basic structure of the inference. So in the intelligent design community with which I'm affiliated, we have agnostic members. They don't know if God exists. They think, however, that the action of intelligence can be detected in natural objects. Two of my very best friends, I won't name them, these were private conversations. They both self-identify as agnostics, but they are active in the intelligent design community because they are persuaded that natural objects can give clear evidence of having been caused by a mind. Now, whether there was one mind or many is an open question. And I think as the intelligent design research community continues to grow, we will see more and more people entering who are not theists, who are not persuaded there is a single designer, but who are fully convinced that it's a worthwhile enterprise to go look for evidence of design should it exist. So I thank the questioner. It's a really insightful question. And I think the move to a single designer is not something that can necessarily be sustained by scientific evidence alone. You need to bring theological arguments to bear. You need to bring philosophical arguments to bear. And I welcome that, right? But I think the scientific enterprise of design detection can still be up and running, even if we leave aside the question, was there more than one designer? So I'm very sensitive to this criticism that intelligent design is begging important questions. And I would not want to foreclose a lively debate about the multiplicity of designers as a possibility. I think that's very important to have that discussion. And I expect it to happen within the next decade. These questions are just too interesting to keep pushing them aside.
Well, Paul, that was the last question. So it only remains for me to thank you again for a great talk and some really good answers to some very interesting questions. So thank you again. And I will hand back now to David Galloway.
Good, David. Well, thank you very much. And let me also add my thanks to Paul. Paul, thank you for a very thought-provoking session, beautifully illustrated talk too, and great input, searching questions from the live audience, which was really great. So thank you everyone for that degree of engagement. One contributor indicated that he plans to go back through the detail and look at the slides again, and I think my friend Asensi won't be alone there. That's the beauty of having it all recorded. Paul has very kindly offered to respond to contact. You can contact him via the website, the c4id.org.uk. That's C, the numeral 4id.org.uk. And you can contact us that way. Make sure that you do register with us so that you receive our emails, and also subscribe to the YouTube channel so that you don't miss any of these videos as they are produced. Now we're planning another monthly webinar in exactly 4 weeks from today. So that'll be the 15th of March, and you can expect email confirmation of yet another world-class speaker on that occasion. And then I can tell you that in April, on the 19th of April, we're going to be hosting Casey Luskin, who's now the new Associate Director at the Discovery Institute, and he's going to be speaking on human origins. And in May, we have yet another treat lined up, and we'll give you more detail of that in due course. So let me just encourage you as we wind up today, make sure you visit our website, make sure that you register your contact details so you don't miss any of these events or updates, and subscribe to the YouTube channel. So I think with that, let me just once again thank Paul for his fantastic input today. We've really enjoyed that, and we'll sign out. That's it until the next time. So everyone, stay safe, and remember to avoid that pesky virus. Thank you very much.
Okay, we are offline.